Students, today we have to talk about the various genes related to the 7 characters which are taken by Mendel for his analysis. Now we have 7 characters considered by Mendel, and each and every character is controlled by a particular gene. Now we have to find out the location of the gene, where are the genes present for these 7 characters. So later it was known the genes for these 7 characters are located only in 4 chromosomes. That is chromosome 1, chromosome 4, chromosome 5 and chromosome 7. So these are the 4 chromosomes where we have the genes for the 7 characters are located. For example, if we are taking the first chromosome, it has 2 genes for 2 characters. If we are taking the 4th chromosome, it has 3 genes for 3 characters. If we are taking the 5th chromosome, it has only 1 gene for a character. If we are taking the 7th chromosome, it has only 1 gene for a character. The chromosomes 2, 3 and 6, they do not have any genes for these characters. They have genes for some other characters, but they do not have genes for these 7 characters. Now what are the genes? So I have taken actually the flower color, the seed color, the pod shape, the plant height, also flower position, the pod color and also the seed shape. I am taking according to the order from 1 to 7. Now let's take character number 1, the flower color. The gene for flower color is located in chromosome number 1. The dominant gene is capital A. So the mutant form, the recessive gene is small. So this is the gene for flower color located in chromosome number 1. So this is gene for flower color A. Now, so it's chromosome number 1. Here the dominant character is purple in contrast to the recessive character white. So purple flower color is dominant over white flower color. The gene for that color is located in chromosome number 1, and that is the dominant gene A that produces the purple color. When it is in mutant form, it produces only the white color. This gene is responsible for the production of anthocyanin pigments which give colors to the flowers. Now let's go for actually the seed color. The seed color gene is capital 1, that is dominant gene I. The recessive gene is what we have the small letter I. It is also located in chromosome number 1. We have 2 types of colors of cotyledons. One is yellow, another one is green. The yellow is dominant over green color. So yellow color is produced by dominant gene I, and its recessive small letter produces the green color. It is also located in chromosome number 1. So this is I gene. I am taking only the dominant gene. So this is the gene for flower color, and this is the gene for seed color. That is a cotyledon color. Now I will go for the pod shape. The pod shape is actually we have one is inflated, another one is constricted. Inflated means swollen, constricted means reduced in size. Now the gene for pod shape, this is the dominant gene capital V. We have the recessive gene small v, and that is located in chromosome number 4. Now this is chromosome number 4, and this is what is called the gene B. I am taking only the dominant gene. The gene for what is called actually the pod shape is located in 4th chromosome towards 1A. And the dominant character is inflated and its recessive form constricted one caused because of the recessive gene. Now the next one, plant height. The gene for plant height is also located in chromosome number 4. And that is the gene LE. This is the dominant gene. So this is the recessive gene. So the dominant gene capital E produces the tall plant, and its recessive form produces the dwarf plant. So here is a gene. I am taking only the dominant gene for what is called the plant height. So the gene for flower color and seed color are located in chromosome number 1. The gene for what is called actually this one, the pod shape, and also the plant height. Now this is the gene for plant height. I made a mistake. Now listen here. It is here the gene for plant height. And we also have the flower position. The gene for flower position is FA and it is a recessive form. Now, there are two types of flowers regarding the position. One is axial, another one terminal. So the axial flower is dominant over the terminal flower. A gene for the dominant character axial flower is located now in the fourth chromosome. Now this is what is called FA. I am taking only the dominant gene. So we have the genes for the pod shape, the height of the plant, and also the position of the flower in chromosome number 4. Now coming to the next one, pod color, that is a fruit color. So we have two types. One is green, another one is yellow. Unlike the seed color, there the seed color yellow is dominant over green, but here the green color pod is dominant over yellow. So it is produced by the gene GB, and this is a recessive gene. Now the gene for pod color is present in chromosome number 5. Now this is GP gene. GP gene, the gene for that is actually pod color, green pod. So G for green, green pod. So green pod is dominant over that is actually the yellow form. Now we have the seed shape. There are two types. One is round, another one is wrinkled. Round is dominant over recessive. Now the gene for actually the dominant character is R, capital R, and its recessive form is small r. The gene for seed shape is located in the 7th chromosome. Now this is a gene. So according to the pictures you can find the gene for what is called the flower color and seed color located in chromosome number 1. The gene for what is called the pod shape, the height of the plant, and what we call the position of the flower located in 4th chromosome. And the gene for what is called the pod color is located in 5th chromosome. The gene for actually the shape of the seed, the seed shape is located in what is called the 7th chromosome. So out of the 7 chromosomes, only the chromosomes 1, 4, 5 and then just 7, they only have the genes for these 7 types of characters. In one chromosome, there are 2 genes for 2 characters. In the 4th chromosome, there are 3 genes for 3 characters. In the 5th chromosome, only 1 gene for a particular character. In the 7th chromosome, only 1 gene for a particular character. I represent all these things in the tabular column. So the chromosomes without any genes for these contrasting characters are number 2, number 3 and number 6. I represented the green color. So these are 3 chromosomes without any of these genes in them. They may have genes for some other characters. So this is the whole, the character, the gene, dominant gene, recessive gene, the chromosome number, where they are located, then the dominant character and recessive character. The inheritance of characters. Mendel crossed and catalogued nearly 24,000, 34 plants for several generations, you know already. And he just published and presented his results in Society, what is called Brunn Society. Brunn Society, the capital city of Austria, of natural history. We know that one, natural history. In, that is 1866, when the book was published, that is Experiments on Plant Hybrids. He just actually crossed and catalogued nearly 24,034 plants for several generations. Then only he came to the conclusion about what is called the genes and their inheritance. Now, we have different genes for controlling different characters. Let us take a gene for flower color. Gene for flower color. I already mentioned the gene for flower color is PGA. You see that one located in the first chromosome, PGA. So actually, if you want to understand the action of gene, you have to know the functions of genes at a molecular level. Then only you can find how far the regulating function of the gene occurs. Now, for regulating the white color flower or for regulating the purple color flower, we have to know the gene functions at the molecular level. Now, it is actually an interesting aspect to study about the function of the genes at a molecular level. Now, the gene for, that is color of the flower, what is called P gene A. This is the gene for color of the flower. So it was normally discovered October 11 in 2010. A team of international researchers about the functional aspects of this P gene A. It is called P gene A, that is gene A present in pea plant. Now, this gene is responsible for encoding a protein. Now, the product of this gene is a protein. The gene A product is a protein. So, this protein is acting as a transcription factor. Transcription factor. This protein is acting as a transcription factor. Only when the transcription factor is active, then there is a formation of the pigment for coloration of the petals. So the pigments are anthocyanin. Anthocyanin pigment. So the P gene is coding for a product which is a protein. It is a transcription factor. When the transcription factor is active, then the gene undergoes expression. So then we have the products and the cyanin is being produced. It accumulates in the petals leading to the purple color. Okay, this is one. Now what is the mechanism? This transcription factor is acting as on-off switch. Acting so, transcription factor so it is on or off switch for gene expression. On off switch for gene expression. When the transcription factor is present, it just actually induces a transcription process leading to the formation of messenger RNA, which results or which are translated to form what is called a specific enzyme. That enzyme is responsible for the formation of the color pigments anthocyanin. Then the flower becomes purple. But in the case of flowers which are white in color, though they have the gene, there is no accumulation of anthocyanin pigments. This is because of the presence of a recessive gene or a mutant gene. Now I will show that one in the presence of a specific picture. Now this is what we have, a DNA segment. So I am taking a DNA segment. We will be studying more about in molecular genetics in zoology. This is a particular gene, a structural gene responsible for transcription, and its expression leads to a particular product. Now, just near it there is a structure. This is a structure. This is what is called promoter region. The region present near the transcription site. So this is a region of initiation of transcription, and nearby we have the promoter, and to this promoter only something will attach that will initiate the transcription process. Now the gene, the one which is to be transcribed, which is to be expressed to produce a product. Now, under normal conditions, there is no gene expression. Then we have a transcription factor. So this is what we have, activating transcription factor. Activating transcription factor. So this factor is acting as on or off switch in gene expression. In the absence of transcription factor, activating transcription factor or active transcription factor, what will happen? The gene undergoes transcription. Now the activation factor, that is a transcription factor, binds to the promoter. Now the promoter stimulates the gene to undergo expression. As a result, what we are getting, you see this one, the messenger RNA. So normally this is the transcription process. The gene is being transcribed. That is nothing but copying of mRNA. So the mRNA is being produced and this one being translated to form an enzyme. This enzyme is responsible for the production of anthocyanin pigment. Anthocyanin pigment. So accumulation of anthocyanin pigment results in purple color. This is a mechanism. So in the presence of activating transcription factor, that is what is happening, the gene undergoes expression and results in mRNA to form an enzyme. The enzyme causes the formation of the anthocyanin pigment responsible for blue, purple, etc. That results in purple color. Now in the absence of this activating transcription factor, though the gene is present, there is no formation of anthocyanin pigments. So the flower becomes white in color. The reason for white color, there is no anthocyanin pigment because of what is called the activating transcription factor or the active transcription factor is absent because it is an on-off switch. It only switches on so that the gene undergoes transcription. Then only we have the formation of mRNA, the enzyme, anthocyanin pigment formation, accumulation to form purple color. So I mentioned already in the case of just white flower, there is no transcription factor which is inactive state. What is the reason for inactive state of transcription factor? Researchers wanted to know how the gene for flower color, that is A gene, is functioning. So they delivered normal gene sequence. So delivery of normal gene sequence A into the cells of petals, not all the cells, into specific cells of petals of white flower by gene-wen method, the method adapted. This is will be studied later under I think cell biotechnology. One of the methods adapted for the transfer of genes into the cells from one cell to the another cell. They are using gene-wen biolistics method adapted. So to produce transgenic organisms. So they delivered a normal gene A sequence into the cells of petals in specific cells, not all the cells, only particular cells of white flower by gene-wen method. Now what is happening in the result? The entered gene expresses itself in the cells because it is a gene expressing, producing anthocyanin pigment in small percentage of particular cells, resulting in purple color in that particular part only. This is because of the expression of the gene and because of the presence of transcription activation factor. The gene is being expressed, resulting in the formation of enzyme which causes the production of anthocyanin pigment, accumulation of anthocyanin pigments leading to purple color in that particular part of the flower. Then, what is the reason for the appearance of white color? Now, the gene A sequence in white flower, if you are taking the white flower, the gene A sequence shows a single nucleotide change. A single nucleotide change. The meaning for that one, a single base change. For example, if you are taking that particular gene, I am taking, this is a gene. In that particular gene, the sequence in one place, one base, what is called guanine or G. I am taking the base G. So this is a normal gene. This is a normal gene. But in the case of mutant gene, what will happen? There is a single nucleotide change. There is one base change. Now, the mutant gene, this is a mutant gene. What's happening in the mutant gene? In place of G, A is present. A change from guanine to there is adenine. This is what's called single nucleotide change or single base change. That results in the formation of a mutant gene. So the mutant gene is unable to just actually produce an active transcription factor. So it makes the transcription factor inactive. So this normal gene makes the transcription factor active, so it switches on the gene expression. But this mutant gene makes the transcription factor inactive. So that there is no gene expression, there is no switching on. As a result, there is no formation and accumulation of anthocyanin pigment. As a result, the flower becomes white in color. Now here the flower is purple in color. Here the flower is what is called white in color. This is because of a single nucleotide change. So that's why we have to study the function of the genes at the molecular level. A single base replacement. G is replaced by A. Guanine is replaced by A. That results in mutation leading to what is called the mutant gene that is unable to synthesize the anthocyanin pigment because the transcription factor form is inactive, unable to cause the expression of the gene. That results in white flower. So this is a mechanism lying behind the gene that is responsible for the production of or for regulating the white color. What is the significance of Mendelian inheritance? So normally Mendel worked at the rules of inheritance. He followed strictly the rules of inheritance and arrived at the correct mechanism of inheritance. Even before the knowledge of the chromosomes, DNA, gene, the cellular mechanism became known. So without all these actually facilities, that is cellular mechanism, knowing the cellular mechanism, gene structure, DNA and then chromosome, he arrived at the mechanism of inheritance without these knowledges. So his meticulous work of inheritance of characters, meticulous work that is giving just actually deep attention, giving great attention to the work, that's called meticulous work. So the insights and meticulous work of inheritance of that is Mendel played an important role. Number one, the development of improved crop varieties. The development of improved crop varieties. So this is a major role played because of Mendelian inheritance. Just it played an important role in the development of improved crop varieties and also revolution in crop hybridization. Crop hybridization. So these are the two important roles, or just actually the effects of Mendelian inheritance. Without any knowledge of DNA, gene, chromosomes and cellular mechanism, he arrived at the mechanism of inheritance, and these mechanisms of inheritance played an important role in the development of improved crop varieties and also revolution in crop hybridization. Artificial pollination or cross pollination by taking the trait that is a flower color. So for that he took, there is a plant with purple flower, a plant with white flower. These were considered as parents. Now step number one, in the purple flower, removal of, that is, actually as per the book I want to write it down, pollen. That is nothing but the male part of flower. Male part of flower is cut off or removed. That is from this purple flower, and this process is being done before fertilization. Before fertilization, that is self-fertilization. Because it's a bisexual flower, there is a chance of self-fertilization. Before self-fertilization occurred, Mendel, what he did, removal of the anther from the purple flower. This process is called emasculation. Emasculation. Now this is the flower without anther. Now step number 2, that is here, I am writing step number 2. I am writing here step number 1. Instead of writing there, this is step 1. Pollen grain or pollen or the anther lobe is removed from the purple flower, making it free from, that is a male reproductive organ, namely the androgen. Now step number 2, a removed pollen. So pollen is taken and transferred to the stigma of white flower. Sorry, mistake, purple flower. Stigma of purple flower. Because purple flower, in which anthers are removed. In which anthers are removed. Not any other flower. The flower where we have removed the anthers, to that flower only we have to dust the pollen grains on the stigma, on the side removed. This is step number 2. So what we are doing, that is a cross-pollination. Maybe what we have the 2 different varieties of the same tree. What is happening? Step number 3. Now the fertilized, the third step, fertilized pistil actually becomes the pod. So formation of the pod from the fertilized pistil, that is actually what is happening after the pollen grains are dusted in order to prevent the cross-pollination from some other flowers. He covered the stigma of the flower by means of what is called actually the plastic covering, so that no pollen grain could reach the stigma from any other flowers. Now fertilized pistil becomes a pod. Now the pods have the seeds. Step number 4. Now the pods become seeds. Pods have seeds. I am writing in this way. Pods have seeds to be planted, ready for plantation, to be planted. All this is not happening immediately, just within 2 or 3 weeks we are getting actually the pollination, fertilization, and then the fruit formation, and then seed formation. Now fertilized pistil becomes the pod. The pods have seeds to be planted. Once the seeds are planted, they germinate into purple flower plant. They germinate into purple flower plant. Now these are all the steps. I can say these are all the steps what he had taken. He had taken a flower color with the contrasting characters. One the purple flower, another one the white flower. He removed the pollen or the anthers from the purple flower and making it only with the pistil, the female sex organ. The process of removal of anther from the plant, that is from the flower, is called emasculation. Then after that he has taken the pollen grain from the white flower and dusted the pollen grains on the stigma of the purple flower. Now it brings about cross-pollination. It results in fertilization leading to the formation of the fruit, what is called actually the pod. The pod contains the seeds. The seeds when planted, when just actually placed in the soil, germinated into the plant, and all the plants resulted from this cross-pollination between what is called the purple flower, white flower plants are all purple color. That indicated that the purple flower color is dominant over white flower color. To understand genetics, we have to know some terminologies in genetics. Number 1, alleles. We know we have contrasting pairs of characters. For example, tall, I am taking dwarf. The tall character is under the control of the gene capital T, and the dwarf character is under the control of gene small t. Now the tall is dominant over what is called dwarf plant. What are alleles? The alternative form of the same gene affecting the same character is called an allele. So capital T is an allele to small t. Or we can say any letter, capital A, is an allele to small a. So alternative form of the same gene, that is A, that is A and small a, these two are called alleles. So normally the individuals, the characters are expressed in different manner due to the existence of genes in alternative forms. So in short, the existence of genes in alternative forms for the same trait are called alleles. The existence of genes in alternative forms, we can also say the versions, different version, alternative forms for the same trait, for the same trait. And this is what is called allele. Alternative form of the same gene. That is, I mentioned the existence of genes in alternative forms. Capital T and small t. Capital A, small a. Capital R, small r. Capital Y, small y. These are all alleles. Now, the next one, what is symbolization? How can you give letters for the genes? That is called symbolization. Symbolization. Now if you make a cross between tall and dwarf, pure breeding varieties. Now we are getting tall one. Tall. So the character which is appeared or expressed in the F1 generation, these are the parental varieties which are pure breeds. The character which is expressed in the F1 generation is called the dominant factor, or dominant character. So, as it is a dominant character, we are taking the first letter of that dominant character as a gene. So, capital T is a gene for, that is a tall trait. And the alternative character, that is a dwarf, as it is not expressed in the F1 generation, it is called as a recessive character. So, we are taking what is called the opposite alternating alphabet. This is called symbolization. For dominant character, that is, we are giving what is called just the capital letter. For recessive character, we are using what is called, that is a small alphabet, and that is small t for a recessive character. This is the second one. The next one, what is homozygous and heterozygous? And for that, we have to know what is called genotype. What is genotype? So it is a genetic constitution of a trait. Genetic constitution of a trait. Genetic constitution of a trait. Or we can say the genetic makeup of a character. Genetic makeup of a character. For example, if you are taking the tall character, so it is controlled by a gene. If you are writing only one letter, that denotes a gene. If you are writing two letters, it is called actually the genotype. So genotype is normally represented by two letters, a single letter being used for a gene. So this is for tall. It may also be expressed as capital T, small t, and both are the genetic constitution of the phenotype. That is what is called, this is the phenotype. I'll tell you what is the phenotype. So capital T, small t, this is one. Capital T, another one. This is what is called the genotype. And if you are taking the dwarf, now we have the recessive character can be expressed only in homozygous condition. Whereas the dominant character can be expressed both in homozygous and heterozygous condition. Now this is what is called the genetic constitution of a trait, and otherwise called genotype. Now we have two different types of genotypes. Number one, homozygous. Homozygous. What is homozygous condition? If you are taking a homologous pair of chromosomes, if the two genes for a trait are similar, then it's called homozygous condition. Now, if you have two dominant factors for tall character, then it's called homozygous tall. If they're taking the dwarf can be expressed only in homozygous condition, and this is called homozygous recessive dwarf plant, and this is called homozygous actually that is dominant tall plant. Homozygous dominant tall plant, this is homozygous recessive dwarf plant. So homozygous is a condition where the two genes are alike, where the two genes for a trait are similar. Now, another one, just genotype, heterozygous condition. What is heterozygous? Now, in the case of Gordon B. plant, what is homozygous? Now, this condition is called heterozygous. If they are taking the homologous chromosomes, if the two genes for a trait are different, located at a particular locus, then it is called heterozygous condition. Otherwise, we can say heterozygous tall. So, we have homozygous tall P plant, homozygous recessive, that is dwarf plant, and also we have heterozygous actually tall plant, tall P plant. Normally, the heterozygous plants are non-pure breeding types, non-true breeding types. So the non-true breeding types are called actually heterozygous individuals. The non-true breeding types are heterozygous in nature. In normal terminology, these heterozygous individuals are called hybrids. What are hybrids? The non-true breeding types are called heterozygous or heterozygous in nature, which are called hybrids. Okay, so that's about a few just terminologies. We have to proceed further in the next class.